Hello and welcome to Forensic Biology for Beginners by Ivy Haw and Caitlin LeMay. We're here to break down the science behind all your favorite crime scene shows. What do you think when you hear forensics? Most people think of the chalk outline or the crime scene tape, but this term actually covers the interdisciplinary world of crime scene investigations. Forensic science, or forensics, is the application of science to the law. It establishes how events took place through the evidence identified at crime scenes, and thereby provides impartial evidence that can be used in a court of law. Some examples of forensic science disciplines are toxicology, the study of the adverse effects of toxins on living things; serology, the identification of body fluids; chemistry, the identification of unknown substances; firearms, the identification of firearms based off of discharged bullets and cartridges; hair and fiber analysis, the comparison of known and unknown fibers or hairs; pathology, identifying the cause of death and the post-mortem interval; and finally, forensic biology, the analysis of DNA. We will focus on forensic biology and its uses for the rest of our presentation.
But what is DNA? DNA stands for deoxyribonucleic acid, and it's the building blocks for all living things. Essentially, it's what makes you, you. DNA is packaged tightly into structures called chromosomes. You have a total of 46 chromosomes, 23 inherited from your mom and 23 from your dad, making a total of 23 chromosomal pairs, as you can see from the diagram on the slide. Each chromosome is made up of numerous genes that code for different traits or functions. These different genes are located at different locations along the chromosome, referred to as a locus, or for plural, loci. At these different locations, you can have different forms. These are termed alleles. For example, this is a gene of color, and it's found at a particular locus on a chromosome. This gene has various alleles that code for different colors, including yellow, blue, green, and purple. As you can see, person 1 and person 2 on the right of your screen have this gene, and person 1 has the alleles for yellow and green, while person 2 has the alleles for purple and green. But why is DNA so important in forensic science? Well, this is because approximately 99% of DNA is the same between individuals, leaving that 1% that's unique to you. Some of these unique areas are located in non-coding regions of your DNA, and within these regions, variations called short tandem repeats are used to create DNA profiles. Short tandem repeats, or STRs, are areas of short repeating sequences of DNA. So, for example, the person on the top and the person on the bottom both have the same repeating unit of A, G, C, and T, but the difference occurs in the number of times the A, G, C, and T are repeated. In person 1, or the person on the top, you can see that it is repeated a total of 6 times, while the person on the bottom is only repeated a total of 4. These differences can be used to make a DNA profile, making these two people distinguishable at this one location. But what does a DNA profile look like? Let's go over some definitions and explore this further. This is an example of a DNA profile. It's actually my DNA profile. In the gray boxes along the top are the locations that I discussed earlier, locations that we use to create this DNA profile, or loci for plural. Each peak on the electropherogram represents one STR. For example, along the first locus, I have 2 peaks. One is 15 and one is 16. This means that on the first chromosome, I have a repeating unit of 15, and on the second, I have a repeating unit of 16. So the 15 allele I've inherited from my mom and the 16 from my dad, or vice versa. When there are 2 different alleles, or STR peaks such as this, it is termed heterozygous. However, if you take a look at this locus, there's only 1 peak. This This means that I have inherited the same allele, so the 10 allele, from both my mom and my dad, and this is termed heterozygous. So now that we sort of understand what DNA profiles look like and what information they can hold, how are they created? These are the steps to create a DNA profile. The first is sample collection. For reference samples, the most common type is a buccal swab, which is a swab from the inside of your cheek. Some other forensic samples, however, could be other bodily fluids that could have been collected from crime scenes. So examples of mucus or saliva or blood, as well as hair or bone, among many, many other types. The second step is DNA extraction and quantification, where we extract the DNA from the sample used in the first step using a variety of different methods, and then we quantify it or identify how much DNA is present within the sample. The third step is DNA amplification. This is where we exponentially amplify the areas of interest of your DNA using PCR, or polymerase chain reaction. Let's talk about this a little bit further. So PCR, or polymerase chain reaction, is a process whereby you preferentially and exponentially amplify your targeted DNA through cycling temperatures. At each temperature, a different process will occur. So the first step is denaturation. This step occurs at 90 degrees Celsius and is aimed to separate double-stranded DNA into single-stranded DNA, as you can see on this slide. The second step is annealing. This step occurs at approximately 60 degrees Celsius and is when we select the areas of DNA that we wish to amplify. The third step is elongation, and this occurs at approximately 72 degrees Celsius, and it's when the template is duplicated, making the single-stranded DNA into double-stranded DNA once again. These steps cycle through until you have the required amount of target DNA to continue to the next step, resulting in amplified DNA. The next step is sequencing. So after the targeted DNA has been amplified, we use a method called capillary electrophoresis, or CE, to sort the now fragmented and amplified DNA. The CE separates the fragments by weight and gives us a raw DNA profile. The last step is to analyze and interpret the raw data from sequencing by converting it into a DNA profile using the software. Through the software, alleles are given true allele calls, which represent the number of repeats in the STR. We also compute statistics and then write a report based on the evidence that has been collected. Next, let's talk a little bit more about statistics and forensics. So statistics and forensics are important to understand the weight of the type of evidence being presented. For example, a DNA profile. Statistics are used to determine the significance of an association. There are two statistics that are normally used. They are called the random match probability calculation and the likelihood ratio. Both of these statistics can give a numerical representation of significance in different ways, which we will discuss in the coming slides. They both also require population databases, which house allele frequencies of the potential alleles within the population. However, we will not be further discussing population database within this webinar. So first, a random match probability statement expresses the chance of a coincidental match with respect to the features within a population. So, for example, some statements you may have heard in your favorite TV show may sound like, the probability of a random individual possessing this DNA profile is 1 in 1 million, or there is a 1 in 1 million chance that a random individual unrelated to the suspect would also share this DNA profile. However, with that being said, random match probability can get confusing, and therefore there's a push towards using likelihood ratios in their place. Compared to random match probability, likelihood ratio is expressed as a ratio of hypotheses, with the top of the ratio representing the prosecution hypothesis and the bottom the defense's hypothesis. Now this sounds confusing, but given the example of John stabbing his brother, the prosecution hypothesis may be that John stabbed his brother while the defense hypothesis may be that John did not stab his brother. Therefore, if the ratio is greater than 1, or there is more evidence pointing towards John stabbing his brother, then the evidence supports the prosecution. While if the ratio is less than 1, or there is more evidence to support John not stabbing his brother, the evidence supports the defense. Now that we understand the basics, let's do a scenario. On Friday, November 13th at 2100 hours, or 9 p.m., there was a break and enter at your neighbor's house. The perpetrator fled the scene before police arrived. However, they left behind a bloodstain from breaking a window. The police have narrowed down the individual to 2 suspects and have received reference DNA samples from each suspect. The question we can ask ourselves here is, can the source of the blood found at the scene be attributed to the suspects? And to which suspect? In the forensic lab, both the samples from the crime scene and the reference samples from the suspects are extracted, amplified, sequenced, and the resulting DNA is analyzed. These here are the results from the crime scene sample. The stars represent areas where the allele is unknown because the crime scene sample was not perfectly preserved. This is something that you come across often for crime scene samples in forensic biology. With that in mind, these are the 2 profiles from the suspects. Are you able to identify if one suspect matches the crime scene sample? Pause the video now to solve the case. So looking at suspect 1's profile here, we can see that at every locus with information, both the samples have the same alleles. And that's compared to suspect number 2, who it seems that only at one locus do the profiles match. With this in mind, we can then disprove that suspect 2 has contributed to the crime scene sample. And then we can also compute statistics to identify the probability that suspect 1 contributed to the crime scene sample. To do this, we use population databases to identify the frequency of alleles of interest within the population. With this information, we can calculate the RMP. I have calculated the random match probability here to be 1 in 13 million. But what exactly does this mean? And do you think that this evidence supports the contribution of suspect 1 to the DNA collected at the scene or not? Pause here to think about this a little bit further. With such a large random match probability, this translates into the probability of a random individual unrelated to the suspect possessing this DNA profile is 1 in 1 million, 1 in 13 million. Therefore, this is strong evidence that the suspect contributed to the DNA profile found at the crime scene, or it's a very unlikely coincidence that has happened. Did you end up getting it right? This concludes the first part of our webinar on forensic biology. Next, I will let Ivy discuss and explore the additional forensic applications of genetics through the fantastic use of SNPs.
Nucleotide polymorphisms, or SNPs, are the most common genetic variant between people, with around 4 to 5 million SNPs in all of your DNA. These SNPs can be used in forensics to identify people in much the same way as STRs are used. Let's try an example. In this example, there was blood found at a crime scene. We analyzed the sample and obtained the profile for comparison, and it does not match the victim. We have the DNA profiles of 3 women who are suspects in this case. Which suspect left the sample at the crime scene? Suspect 1, Suspect 2, or Suspect 3? Pause the video now and make your selection. If you guessed suspect number 2, you are correct. In real-life cases, forensic scientists look at many more loci to ensure that the profile is truly unique to the individual that it belongs to. SNPs in forensics have several applications in addition to individualization. They can be very useful in genealogy as well as forensic phenotyping. Forensic genealogy is based on the fact that families share some of their DNA. In this family, we're interested in the yellow circle. The amount of DNA shared between the yellow circle and their siblings and parents in blue is 50%. The amount of DNA shared between the yellow circle and their aunts, uncles, and grandparents in purple is 25%. And the amount of DNA shared between the yellow profile and their cousins in green is 12.5%. Forensic genealogy is the process where law enforcement compares more than 600,000 SNPs to large public population databases, looking for relatives of the mystery profile. In this scenario, the yellow circle is the unknown profile that will be uploaded to the large population database, and the green profile is a cousin that has done a test like 23andMe and has uploaded their resulting profile to a public population database. When police upload this profile to the database looks for how much of the DNA is similar to other individuals within the database. It will then return profiles that share DNA and can identify a third cousin or closer relative. In this case, there is an individual that is found to be a possible relative of the yellow circle. Law enforcement can then use this information and investigate the relationship accordingly. Another application of SNPs in forensics is using unknown samples from crime scenes to predict the physical characteristics of the individual that left it there. This is done by using SNPs and genes that dictate physical characteristics like eye and skin color to predict the appearance of the perpetrator. To do this, SNPs for all of the traits are combined and put through an algorithm to predict the profile of the individual that the sample belongs to. Here you can see 3 individuals who were participating in research testing the accuracy of the predicted traits. They each provided a sample for sequencing to predict their facial structure, skin color, and eye color using algorithms. These are the results. These algorithms aren't perfect, and there are environmental factors that play a role in this, but this could provide enough of a resemblance to aid in an investigation. In this webinar, we've learned what forensics is, what forensic biology is, what a DNA profile is, how to solve a case with DNA evidence, what SNPs are, how forensic genealogy is done, and how forensic phenotyping is done. Thank you for watching. We hope you learned lots about forensic biology. If you have any questions, please put them in the comments below. Thank you.